Hello students, you are welcome to our class CRP 502, Plant Growth and Development. This is a program or a course for those offering BE Agriculture at the 500 level. Very interesting. It's going to talk about nature as it relates to plants and its growth, those factors that affect them, and then other issues that we discuss. I'll be your facilitator, and my name is Godwin Alhassan for the next semester. Feel free to ask questions and enjoy the course. God bless you. Mm, Today we are going to on a normal topic, plant growth and development, which for my introduction I told you is a 2 credit hour course, and today we are going to discuss. Today we are going to discuss on that issue again, plant growth and development, the CRP 502. As I told you, it's a 2 credit hour course, and I believe that from the factors and other issues we are going to discuss during this program, we are going to enjoy this course. And my name is Godwin Alhassan of the Faculty of Agricultural Sciences, National Open University of Nigeria. Our topic, first topic for this course is seed germination and dormancy. And by way of introduction, I want to tell you that seed is the beginning of the next generation of plants. And again, seeds have to germinate in order to get established to produce the next set of other set of seeds. Nature has it that there are different periods allowed for seeds to initiate the process of germination. The objectives for this topic are as follows. 1. We have to understand what is seed germination. 2. Know the conditions or factors necessary for germination. And number 3, understand the meaning of seed dormancy. And then we have to equally know the importance of the seed dormancy and then how to break seed dormancy. Let's look at seed germination. As I earlier told you, seed contains the embryo, and the embryo is a new plant in miniature. It contains enough food results to sustain the growing seedlings until it establishes. Germination commences with the uptake of water under favorable conditions like temperature, and then equally to be completed with radical extension that will penetrate the structures that surround the seed. Let's look at the phases of germination. There are 4 phases. The first one is a rapid initial uptake of water and nutrients. The second one is a plateau stage that is going to level up. It seems as if to say it has ceased, and then further uptake of water will now make the germination to continue, and then finally the germination is completed. Today we are going to on a normal topic, plant growth and development, which for my introduction I told you is a 2 credit hour course, and today we are going to discuss. Today we are going to discuss on that issue again, plant growth and development, the CRP 502. As I told you, it's a 2 credit hour course, and I believe that from the let's look at conditions necessary for germination. We have the external factors such as water, oxygen, and a suitable temperature. Water that is adequate but not waterlogged, enough oxygen for respiration, and then good temperature of between 5 to 30. I do temperature range for seed germination. Under a very cool temperature, it will not germinate. Then we have internal factors, which is dormancy itself. When the crop is under a normal dormancy period, germination will not take place. Then this one will lead us to the next slide, which is seed dormancy. There is no one way definition of the phenomenon called seed dormancy. But by simple explanation, dormancy is the failure of an intact, viable seed to complete germination under favorable conditions. We have types of dormancy. We have two types of dormancy principally. We have the coat-enhanced dormancy and the embryo-enhanced dormancy. Let's look at the importance of dormancy. Is dormancy important in our study of nature? Yes. Dormancy is important because it allows seeds to pass through drought, cold, and other unfavorable conditions. It's equally important in the area of dispersal of seeds, and dormancy encourages under favorable condition. Dormancy should be able to be broken and germination will take place. And then dormancy is equally essential for seed storage. How do we break dormancy in seeds? There are many processes. Development of growth hormones, the counter growth inhibitors. Number 2, leaching of germination inhibitors. Number 3, the maturation or after ripening of embryo. And number 4, weakening of impermeable and tough seed coats by microbial action, abrasion, passage through digestive tract of animals, etc. They will continue in continuation of our seed dormancy. We have what we call the scarification, the stratification, and counteracting inhibitors. There are some inhibitors that will counteract germination. We have potassium nitrate solution. We have some growth hormones like gibberellin and thiourea. And then we equally have what we call the shaking under pressure. But by the time you vigorously shake your seed under hydraulic pressure, you are likely to weaken the seed coat, and that will allow moisture to penetrate and germination dormancy will be broken. At this end, I want to thank you for listening, and we wish that we meet next at the same time next week. God bless you.